Hello, welcome back to another mini science session, and today we are going to talk about the role and structure of bile salts. Alright, let's go. So bile salts, like cholic acid, they have both the hydrophobic and hydrophilic phases, okay? So same as what we talked about earlier, they have where is my marker? Here you go. They have, whoa, not that. This one. They have, nice, okay. They have hydrophobic and hydrophilic phases. Nice. And that, what's the term for that? That amphipathic nature will allow them to solubilize the digestion products and transport them across the intestinal epithelium. So again, the bile salts are amphipathic, meaning they are hydrophobic and hydrophilic side, loves water and hates water, and that will allow them to transport products across the intestinal epithelium. And the micelles are not spherical, but they form dynamic, elongated shapes that embed the lipids inside, okay? And the micelles would deliver lipid contents to the brush border of the intestines, where the fatty acids are absorbed. Okay, so what am I talking about? Because in the previous video, let's just write a mini map here to orient ourselves where we are. So we talked about how if we want to digest TAGs, triacylglycerols, we start off with, okay, if we eat meat, this will be meat. This is steak. This is, what is that? That's steak. If you have steak, then you eat it, you digest it using your mouth. This is your teeth. And then it becomes, well, that digestion is like, that's mechanically in the mouth and also in the stomach, where the lipids are then emulsified into droplets. So right now it's broken up, and then it goes through the stomach. And now it looks like mini droplets. Emulsion. And this is still unstable, okay? And that's not yet digestible. So in the gastric phase, that will target the SN position of the tags. So gastric phase, yes, it's a, the science, it is here, even in the science season. Welcome back, Zimbilos. Yeah. Okay, next, after the stomach, the lipases begin to hydrolyze the ester bond at the carbon 3 of CAGs. By the way, you can change your hats here. Hello. Let's see. You can change your hat using this one, hat. Like if you type !hat. There you go. My character will drop by soon. Okay, so you go stable emulsion. You allow the further enzymatic process. Yeah, your hat changed into pizza. And so you have the emulsion droplets dropping into the small intestine. So now they will go into the small intestine. And there, in the small intestine, two things happen. Oh, you like pizza. Which flavor? Speaking of which, this is also digestion, so think about it as pizza instead of steak if you want. So in the small intestine, you have the pancreatic lipase. Not cheese pizza. No pineapples. pancreatic lipase. I am doing digestion. You have pancreatic lipase, which will, what's this called? Act on the TAGs, and that will cleave the bond, the ester bond at the 1 and 3 positions. And then you have 2 mg plus free fatty acids. Okay? And the second one is bile acids secreted by the liver would solubilize the lipids into mixed micelles. This is how it looks. So you ate your student exams. So business student, it's okay. Yeah, thank you for dropping by. Yes. Yeah, you have a tag steak, you eat it, you break it up, it is unstable. It's now in your stomach, and then pancreatic lipase and bile acids would happen. Pancreatic lipase, you have the TAGs. It breaks it up into 1 and 3. Wait, it breaks it up into hello, welcome to the stream. You can type and the character would fall, and you can change your hat into using the exclamation hat. Thank you. Yes. So again, steak. You eat it. Teeth broken up into small pieces goes into your stomach, and the stomach, it's still in like big liquid forms. No, it's still unstable. So in the stomach, you have the pancreatic lipase and bile. Pancreatic lipase would, I still don't like that. Pancreatic lipase would break it up into 2 mg, 2 monoacylglycerols, and also into free fatty acids. And then you have bile, which forms it into micelles. So that's where we're at right now. Bile salts have cholic acid. There you go. It changed into one of the emotes. Perfect. Bile salts are amphipathic in nature, and that's why they can stabilize and carry stuff. Okay, now that they are delivered into the small intestine, that is where the fatty acids and the mgs are absorbed. How the newly synthesized tags, well, they get reassembled. In the small intestine, they get reassembled into tags. But the newly synthesized tags are packaged to form chylomicrons. Okay? So the chylomicrons enter the lymphatic system. Lymphatic system, and that is where they will eventually reach the systemic circulation. So after they leave the small intestine, after the chylomicrons leave the small intestine, what happens? Well, they just go to the different organs, like the heart, the veins, heart, lungs, arteries, liver. So, hello, Planet Flabit. No, you are not late. We just started a mini review. Last time we stopped around here in the micelle formation. But yes, and then after the bile salts form the micelle, pancreatic lipase will form the tags into 2 monoacylglycerols and fatty acids, and then those get delivered into the small intestine, and then they get reformed into TAGs, and then you make chylomicrons. Once you are at the chylomicron stage, what happens? You go through the lymphatic system, and then the veins, and then the heart, and then the lungs, and then arteries, and the other tissues, and then you have the liver at some point, because you remember veins carry blood to the heart, And then the heart and the lungs, we know that they would just exchange stuff. And then after that, arteries carry blood away from the heart to the body parts. And then after that, the liver. So it's the liver is the last organ that they encounter, and only little, because you have the absence of lipoprotein lipase on liver endothelial cells. So that's why that's the last one that they encounter. Then how are tags and chylomicrons, like what happens in the blood? So the TAGs, or triacylglycerols, and chylomicrons, they are hydrolyzed in capillaries of target tissues. So what does that mean? Well, I think I have to write it out. But essentially, you have the lipoprotein lipase, right? It is activated by the ApoC. Wait, let's just draw it out. Drawing. Let's draw it here. Erase and draw more here. Okay, so you have the TAGs. What's up? Nice star on your name, Mr. Big Balls. How do you put the star on your name? Is that like an emote? Wait, so you have the chylomicrons. Spelling of these. Then you have the lipoprotein lipase. And then you have the FAS. At some point you get FA, and then you have albumin floating here as well. Okay, so the lipoprotein lipase would cleave the TAG into fatty acid and monoglycerol from changing your... Oh my goodness, I did not know you could do that. I'm gonna try that. Putting 2 sword emoji. TAG, and then you have lipoprotein lipase. So what was the thing that converts triacylglycerols into monoglycerols and fatty acids? Lipoprotein lipase. LPL. Okay, good. And after that, they are taken up by the cells. So how for fatty acids to be used in metabolic reactions, they must first be activated into fatty acyl-CoA, and this occurs on the endoplasmic reticulum via 2 steps, a 2-step reaction, I mean. So let me just scribble that out here. So I think I have to write the TAG and lipoprotein lipase into monoglycerols plus fatty acids. That one is good to remember. Okay, for fatty acids to be used, they must be activated into fatty acyl-CoA. So from fatty acids into fatty acyl-CoA. It has 2 steps, that's why there are 2 arrows. Fatty acyl-CoA synthetase. No, it's okay. This is about eating food. If you eat food, you can understand this. Slowly but surely. Yes. So fatty acyl-CoA. We're talking about how after you ate steak, it's broken down. How does it go from steak into your bloodstream and to the liver? Fatty acyl-CoA. So what's in the middle? You have an intermediate. The process would consume ATP into fatty acyl-CoA at some point, CoA and AMP. And all of these happens where? In the ER, endoplasmic reticulum. So that's why when y'all were talking about cells in class, that's what they mean by the lipid stuff happens in the ER. Yeah, they're connected. Amazing. Oh, and by the way, the fatty acids would enter pathways like beta oxidation. Hello, George Rojas. Welcome back to the stream. We're talking about how if you ate steak, what happens after you chew it? It goes to your stomach, and then now it's in your bloodstream. We're now in the endoplasmic reticulum section. Yes, there's glycerol. Oh, we talked about this. Okay, adipose tissue. We know how what adipose tissue is, right? So that's the fat that we know. It's in the subcutaneous layer and abdominal cavity. It is also the primary site for long-term energy storage, and it contains fat droplets, mostly composed of triacylglycerol. So that's why when they say, oh, fat and sugar, those are energy storage. It's also not good if you have too much of those stored. But generally, they can supply energy for weeks, especially during fasting. Around 25% of the body weight is typically fat. And during the fasting state, the adipose tissue would use the tags to release fatty acids for use by tissues such as the skeletal and cardiac muscle. Okay? And while the skeletal and cardiac muscle would store tags within the lipid droplets, they do not actively synthesize tags in the fasting state. Meanwhile, our intestines do that. Okay, they reassemble the tags from monoisoglycerol and dietary fatty acids. So we mentioned that before. Wait, I will erase first. So, we mentioned how, oh, it's so fun, a lot of the mini bobbleheads are on the screen. Nice. Okay, so we talked about how the skeletal and cardiac muscle, both are muscles. Muscle. They store tags. You have tags, right? But they can't synthesize tags. So that's why the intestinal mucosa would reassemble tags make tags from monoglycerol and dietary fatty acids and packages them into chylomicrons. Yay! And the liver is not a good site for export, and its role is more prominent in endogenous lipid metabolism. Okay? Now, let's talk about feeding and the fed state and the fasting state. Fed state is, let's see, in the fed state, where? Fed state just means if you ate. Fed and fasting. And the use of fatty acids. So in the fed state, fatty acids from food is digested and absorbed through blood and stored as tags in the tissues. But in the fasting state, they would shift to using. Here is using the tags. So one will promote storage, one will promote use. Of course, because if you are well fed, you don't need to use them up. You need to store them, but for fasting, you need to use what you have because you're hungry. You will die if you don't get enough. So, maybe not die, just faint. Okay. How do you mobilize fatty acids when you're fasting? Let's see. Erase. How do you use it? So you have 3 enzymes. Here. Let's write them out first into a mini map. You have tag, You have the adipose triglyceride lipase, ATGL. I think after ATGL, adipose triglyceride lipase. It converts TAGs to DAGs, diacylglycerol. And okay, because this is triple, now it's di 2. Okay, so TAGs into DAGs using adipose triglyceride lipase. Lipase just breaks it down. Now from DAGs into MAGs, you have the hormone sensitive lipase. And then from the and then the MAGs to glycerol and fatty acid, you have the monoglyceride lipase. Those are just 3 things. Okay, 3 enzymes. Adipose triglyceride lipase would convert TAGs into DAGs, dry into di, hormone sensitive lipase di to MAGs, and then monoglyceride lipase MAGs into glycerol and fatty acids. Beautiful. And then the fatty acids will now be delivered to the bloodstream for release. Perfect. See, just arrows. Nice arrows. Okay, so now, once inside the cell, the fatty acids must be converted to fatty acyl-CoA or by the acyl-CoA synthetase, right? Okay, so now let's try that. We talked about, because they flow through the bloodstream and goes inside the cells. So now they're in cells. Fatty acyl-CoA. Oh, I think we can just write. No, I'll write it here so that the map is still. I'll try to change my hat to hat. Exclamation hat. There you go. Because mine is a crying emoji. I want it to be the dab emoji. Fatty acid, you're inside the cell. You need to convert the fatty acids into fatty acyl-CoA. Oh, it is now the happy emoji. That's fine. Fatty acyl-CoA by what? The acyl-CoA synthetase. Okay, that's good. Now we have the fatty acyl-CoA synthetase. The short and medium chains. Ooh. Okay, this is a fork. The short and medium chains. Short and medium would fuse directly into the mitochondria. What is the mitochondria? Say it with me. Mitochondria is the powerhouse of the cell. Dab. Okay. Mitochondria. So the short and medium fatty acids would fuse directly into the mitochondria. While. Yes, we said the thing. I want to type it. Mitochondria within capital letters. Mitochondria is the powerhouse of the cell. Yes. Hello, great teacher. Good timing. We were talking about how the mitochondria is the powerhouse of the cell, and that's also where the short and medium chain fatty acids would go. It would just fuse directly into the mitochondria, while the long chain fatty acids needs the carnitine shuttle. It needs the carnitine shuttle. Okay? So both of them end up in the mitochondria, but one is just direct. This beautiful artwork you see here, you can just post it in a museum. Okay, mitochondria, this one is L-carnitine shuttle. But then essentially they are now here in the mitochondria. And once inside, that is where the beta oxidation happens. Okay, and beta oxidation will lead to the acetyl-CoA for what? For the TCA cycle and ATP production in the electron transport chain. See, and this is all because you ate steak or a burger, something with fat. Okay, so again, as a mini review, we eat. If you ate steak, let's say you ate something. Oh no, this part is when during fasting and we're breaking it up. Okay, so TAGs, the triacylglycerol gets broken down into diacylglycerol using what? Using the adipose triglyceride lipase. And then from the di into the mono using hormone sensitive lipase. Monoacylglycerol into the what? Glycerol and fatty acids using monoglyceride lipase. After monoglyceride lipase, the glycerol and fatty acids, you have that one. Fatty acids would just flow through the bloodstream, gets delivered into the cells of specific body parts or organs. You have the fatty acids would then get converted into fatty acyl-CoA using the acyl-CoA synthase. After that one, you get the small and medium chains would get delivered into the mitochondria just by fusing, but the long chains would need the L-carnitine shuttle or the carnitine shuttle. Once you are inside the mitochondria, you undergo beta oxidation. After beta oxidation, you now have the acetyl-CoA, and that's where that's a very familiar word because that's the same thing that goes into the what? The TCA cycle. TCA or Krebs. It has 3 names. TCA Krebs. That's the tricarboxylate, right? And another one. Yes. Oh, that is true. It's okay. Just flip it 90 degrees. No. Do not break your neck. It's okay. Now, we mentioned how the long fatty acids must be transported for beta oxidation also, right? Carnitine shuttle will help. So, but how does that happen? This is the long one. First, they must be converted into acyl-CoA while it is in the cytosol. And then that's where the CPT1 happens. We could write it actually. Should we? Okay, let's try that. I'm gonna erase this. The big beautiful map. Let's erase. Okay. Can't believe I'm writing the carnitine shuttle. Here. Okay, there you go. So, let's say you have... This is the cytosol. Now, this is the outer mitochondria membrane. Outer mitochondria. I should just write it. Outer mitochondria membrane. And then you have this. This is the inner mitochondria membrane. Inner mitochondria membrane. And then this is just the intermembrane space. And then after that you have the actual matrix. Alright, let's try it. Okay, you have the fatty acid. Let's write L, that's the long fatty acid. Okay, you are first converted into fatty acid, acyl-CoA. In the cytosol, you have the acyl-CoA synthetase. First, your long chain fatty acid. Yes, long chain fatty acid into the fatty acyl-CoA. Okay, just remember if this is during the fasting state. After take note, you need ATP here. ATP into AMP. So now that you have that fatty acyl CoA, it now goes inside the cell. Fatty acyl CoA. And then you have CPT1. This is CPT1. CPT1. The carnitine palmitoyltransferase 1, which is outside in the outer mitochondrial membrane, it would form acyl carnitine, fatty acyl carnitine here, like that, fatty acyl carnitine. Note that it would also use carnitine. I'll tell you where that comes from later. Now that you have fatty acyl carnitine, using the carnitine, there's a lot of words here, I apologize. Using the carnitine acyl carnitine translocase, the fatty acyl carnitine would be teleported here, and then you have a CPT2 which would make it into the fatty acyl CoA for beta oxidation. So essentially this is just how to teleport or transport the long chain fatty acids from the cytosol and inside the actual mitochondria. So it gets converted into fatty acyl CoA a little bit into the intermembrane space, and after that, with carnitine, it becomes hello Landor. Welcome back to the stream. We are talking about how in the fasting state you have fatty acids or triacylglycerols and then into fatty acids, and now we're trying to put it into the mitochondria. Where what is the mitochondria? The powerhouse of the cell. Now, okay, so far we talked about generating energy. We talked about moving fatty acids out of the adipose tissue for use by tissues in need of energy. And this is also, next could be the generation of energy from fatty acids in the tissues with mitochondria. Ooh, this is so nice. There's so many bouncy people. Thank you for chatting in the stream. And yes, you can change your hat by typing !hat. Mine, I think, is the happy waving emoji right now. Yay! Okay, now let's jump into talking about the beta oxidation. Because right now, we have the beta oxidation happening. Let's go. Erase. Here. Yes, you can change your hats. And take note, I think after you change your hat, there might be like a 5-second wait before you can change your hat again. I don't know. I think that's just how, so that it won't crash. Now, let's talk about beta oxidation. It will convert the fatty acyl CoA to acetyl CoA. Okay? The acetyl CoA would feed to the TCA, that TCA cycle. And that is also where, these terms, the burger, you got a burger? Ah, yes, there's a pizza too. Burger, pizza, apple, hat. Oh, take note, I'm not sure if you all noticed, but whenever you type in chat, your mini icon would have the ... So it'll show if you're talking. Yes. But I don't think it will. I think the exclamation point hat would just change the emote, but when you're talking normally, that's when the ... will appear. Okay, the acetyl CoA would then, reacts with we know the oxaloacetate. It's usually somewhere here, oxaloacetate, to form energy, generate energy, because that's ETC after. See? Beautiful. It's all interconnected. Yes. Okay. So, the oxidation of palmitic acid yields, like that's a 16 carbon saturated fatty acid, right? It will yield an impressive 106 ATP because you use up, like after that, you use 2 ATP for during activation, 28 ATP are produced from 7 rounds of the beta oxidation. And then you get ATP, 80 ATP come from the oxidation of 8 acyl. Essentially, the TAGs, the triacylglycerols that are inside your body. You have apple. Yes, apple. The TAGs. Who has the Sherlock Holmes hat? That is cool. Oh, Planet Planet has it. Nice. I didn't even know that there was a Sherlock Holmes hat. Okay. Essentially, if you are fasting, the TAGs will be used as a protein source. But one might ask, can proteins be used as the energy source? Well, we have to make it's complicated. It's not a primary fuel source. Yeah, no, I think you had the Sherlock Holmes one. Yes. Okay, so in state of fasting or starvation, amino acids can also be used as like a primary fuel source. Not primary, but as a temporary fuel source, because, for example, here you go. You have amino acids from the diet, excess amino acids from the diet, excess, can be converted into glucose or fatty acid for storage. And during prolonged fatty acids, prolonged hunger, the carbon skeleton of fatty acids are converted into acetyl-CoA, which can also enter the TCA cycle. But why is that? But that's kind of not good. Why? Because it also generates like a bad thing called ammonia, and that you need to detoxify that into urea, which is energy. You need a ton of energy for that one, and you already don't have enough energy. So that's why it's not ideal as a fuel source. So again, our body is amazing. You can have lots of things. There's a lot of things. Lots of things are going on right now. Even if we're just like sitting here. Okay, now let's talk about ketones. How many is it? 1, 2, 3, 4, 5, 6, 7, 8, 9, I think. 9 bouncy thingies. It's nice. Yeah. Okay, so in states of low insulin or high glucagon, that one is the fasting, right? Here. Low insulin or high glucagon, such as starvation or fat or type 1 diabetes. Okay, cat, that's good. So yeah, during starvation, fatty acids are mobilized rapidly from adipose tissues, and the liver would take up the fatty acid, convert them to acetyl-CoA. But when the hepatic energy needs are met, the excess acetyl-CoA could be diverted and made into ketone bodies. And this shift would ensure continued energy supply to tissues like the brain. This is the brain. Because and that's important because it can't use fatty acids. Note, only the mitochondria in the liver cells could convert can synthesize ketone bodies, okay? Because you have HMG CoA lyase, and that's only in the liver mitochondria. So again, only the mitochondria in the liver cells can make ketone bodies because it has HMG CoA lyase. So yes, 3 acetyl CoA would form HMG CoA, and it is then cleaved to produce acetoacetate. A lot of acetones. And then after that one, the ketone bodies could have 2 fates. Okay? Let's erase ketone bodies. So we now made ketone bodies inside the liver. What happens to that? We are now in the liver, mitochondria, and we have ketone bodies. Okay? Ketone bodies like acetoacetate. 2 fates can be made into acetone, which is exhaled or excreted, or it can be reduced to beta hydroxybutyrate via the beta hydroxybutyrate dehydrogenase. And that will also depend if you have enough NAD plus or NADH. That's also reversible, so the arrow goes both ways. And the ketone bodies can just keep on flowing to the bloodstream to supply energy. And tissues outside the liver, such as the skeletal and the heart muscle, are the primary consumers of ketone bodies during early starvation. And as fasting continues, the brain shifts to using ketone bodies to spare glucose. Okay? And in the mitochondria of those tissues, the brain and the heart, they're always partners. This is the heart, this is the brain. Oh no, we're talking about skeletals. So it's just the heart and the muscle. This is a muscle. In the mitochondria of those tissues, it's first, the beta hydroxybutyrate is first reformed into acetoacetate, and then acetyl-CoA, and then after that, acetyl-CoA, just so you can use it for energy. And that's also where like ketogenic diets come from. Like, it consists of high fat, moderate protein, and very low carb intake. It mimics a fasting state by promoting ketosis, aka the part where the liver produces ketone bodies as the main fuel. This diet is usually used to control seizures in epilepsy and kinda have been shown to support weight loss by encouraging fat burning, but it has to really be monitored carefully because it can lead to nutrient deficiencies and could mimic type 1 diabetes because uncontrolled ketosis can be dangerous. So yes, and then take note that when you so in the body there is like the changes across fed, fasting, and starvation rates, because as the body transitions from fed to fasting and eventually starvation, the blood concentrations of glucose drop while the levels of free fatty acids and ketone bodies rise. And initially, the brain relies on glucose, but during prolonged fasting, it will adapt to ketone bodies to prevent to preserve the muscle protein. So this shift in fuel usage would reflect hormonal changes. So let's draw that out. We have a graph. We can have a graph. Serious science and math. Okay. You have this. Okay, this is hours of starvation. Either you forgot to eat because you were studying for so long, or you were playing Minecraft for 12 hours. And then this is relative change of y-axis is relative change of values. So let's start with the blood glucose. The blood glucose is this one. Blood glucose. That's sugar. Now you have the plasma-free fatty acids going like this. Fatty acids. And then you have the ketone bodies going like this. Ketone KB. And then you have the glycogen. Let's make it gray. Glycogen would be going from here to here. Because there you go. Then you have the plasma insulin. Something like this. Plasma insulin. And then the plasma glucagon, which is going the opposite way. Because insulin and glucagon kind of work in opposite ways. So just mini questions. How is lipid metabolism integrated with glycolysis, the TCA cycle, and amino acid metabolism? Well, the lipid metabolism is interwoven with the central carbon metabolism through shared intermediates, because the glycerol from triacylglycerol breakdown is converted into G3P and also the DHAP, the DAP, G3P DAP. We wrote these down during glycolysis and they feed directly into glycolysis. Fatty acid beta oxidation also produces acetyl-CoA, which enters the TCA cycle. And then amino acids like glutamate and alanine, which are transaminated to form pyruvate, and pyruvate also goes inside the TCA cycle. And what is the biochemical significance of pyruvate? Oh, we talked about this. Beta oxidation breaks down long chain fatty acids, 2 carbons at a time. What's the function of carnitine shuttle? Ooh, that's it, because the long chain fatty acids can't just diffuse in, so they will into like different parts. Just use CPT1 from on the outer mitochondrial membrane, CPT2 as well, until you have the acyl-CoA. How does the body regulate the switch between lipid synthesis and breakdown? Well, that is because in fasting or starvation, they will activate like acetyl-CoA carboxylase and just suppress hormone-sensitive lipase. Remember the steps from TAG into the FA and MG. Oh, and FA and other stuff. What is the fate of fatty acids during prolonged fasting? Well, during prolonged fasting, adipose tissue breaks down the TAGs into glycerol and free fatty acids. Glycerol is used in the liver for gluconeogenesis, while fatty acids is for during beta oxidation. Structure of fatty acids. How does the structure affect its function and physical property? Well, the chain length and degree of how much it is saturated affects the melting point and membrane fluidity and metabolic rate. Saturated fatty acids do not have double bonds. They pack very tightly and are solid at room temperature. Unsaturated fatty acids, especially in the cis configuration, introduces the kinks that lower the melting point and increase the membrane fluidity. And what distinguishes the essential fatty acids from the non-essential ones? Essential cannot be synthesized by the human body because we lack the enzymes to introduce the double bonds to them. So that's why they should be obtained from the diet, and that's also because they are critical for brain development and membrane integrity. So that's why eat fish and stuff like that. What happens to chylomicrons after they deliver dietary lipids to tissues? After they are hydrolyzed, the chylomicron remnants are rich in cholesterol and phospholipids, right? Those would be taken up by the liver via the receptor mediated enzyme, what's this called? Endocytosis. And the liver recycles their components for bile acid synthesis. Okay, I don't think we brought this up before, but how does, if you have an odd chain fatty acid, how does that connect lipid and next emote should be mitochondria. That's true. The emotes, I think, maybe the 6th one if we have a chance. I'm debating whether what do we have right now. I'm not sure if we have the I could release a poll, but there's the 12 photos. I will recheck which one. The light stick emote, I really would like it at some point. It's still being cooked. Haha. The priority was the dab emote. Ooh, and the salute emote. Yeah, salute and light stick emote soon. Hopefully. Okay, so if you have odd chain fatty acid, that's the weird one because unlike even chain fatty acids, which produce only the acetyl-CoA, odd chain fatty acids would also yield propionyl-CoA as a final beta oxidation product. Okay? And take note, breadstick emotes. Thank you for the suggestion. Well, the light stick emote is still being made. And while the propionyl-CoA is converted, the propionyl-CoA, which is what happens if you have the odd one, it is converted to succinyl-CoA via vitamin B12 dependent pathway. So that's why please drink your vitamins, you need B12. And that the succinyl-CoA will enter the TCA cycle and support gluconeogenesis later on. So yes, it really helps. So now we talked about fatty acids and those chains. Ooh, now let's talk about the citrate-pyruvate cycle. Let's erase this. Colorful scribbles. Citrate-pyruvate cycle is next. Womp womp. Okay, you have citrate-pyruvate. Why is that blue? It's okay. Pyruvate cycle. That's actually kinda nice color. So the citrate-pyruvate cycle is critical, right, for fatty acid synthesis because we are now in the making of it. Last time we broke down the fatty acids. Now we're making fatty acids. So citrate exits the mitochondria. Mitochondria is the powerhouse of the cell. Okay, this is the mitochondria. Let's make it green. This is the mitochondria. Every time we see or hear mitochondria, we have to say the line. The line. Okay. Citrate exits the mitochondria. Citrate exits the mitochondria and is cleaved into acetyl-CoA, and oxaloacetate. The obligatory line, yes. Even if you don't type it in chat, just say it out loud. But it would be funnier if suddenly it's like boom, 10 mitochondria is the powerhouse of the cell in capital letters. Mitochondria is the powerhouse of the cell. Okay. So the citrate-pyruvate cycle really links the glucose metabolism with lipid synthesis, because they're all connected. They're connected. For now, let's make a mini drawing, okay? And the drawing will have glycolysis, pyruvate, and all that stuff. Let's draw. Nope. I need my marker. Okay. This one, we have the cytosol, right? Cytosol. And then we have the mitochondrial matrix. Hello, cellophane phone cell. Right now, not yet. Mitochondrial matrix. We're drawing a different one. I drew mitochondria twice. Matrix. Oh my gosh! Thank you, Marley. That's my first tupa. Thank you. Arigatou gozaimasu. That is so cool. Let me heart that one. That is crazy. And this is all because of mitochondria. Thank you. Mitochondria is the powerhouse of the cell. Okay. So we connected from glycolysis. I still can't believe that's my first tupa. Yay! With smiley face. So yeah, we have cytosol, Shinjuku. Oh, how is the, wait, yoroshiku onegaishimasu. Wait, what, how's the weather? Is it tsumeta? Is it warm there? Atatakai? How is it in Shinjuku? Yes. Oh, by the glycolysis. Let's start with glycolysis to pyruvate. It is cold and dry. Yes. Ooh, hello, Lendor. Lamel, the cute little heart emoji. Oh, pyruvate here. Where's my marker? Okay. We have the membrane. Let's draw squiggly lines just to make it clearer where we are. Okay, these are the hydrophobic heads and the hydrophilic heads and hydrophobic tails. So the pyruvate, and we have a protein here, pyruvate to pyruvate. That's how it pyruvate. Cold and dry. Is it different compared to like the weather? Yes, it is my first super chat ever. Thank you very much for that one. And I like how it's in yen as well. Japan, let's go. Glycolysis to pyruvate and go to pyruvate again. Now, this is while in the mitochondrial matrix, mitochondria into pyruvate into oxaloacetate, and then oxaloacetate and acetyl-CoA. This is just connecting them. I wish we had a better pen because this is a great figure. Oxaloacetate. I will try my best to draw it. And then you have citrate. Citrate into isocitrate and all that. See, that's why TCA. So now you can see how glycolysis happens in the TCA. And glycolysis happens in the cytosol. TCA happens in the mitochondria. Now, what happens here? I left a lot of mini spaces. From the glycolysis, we have, what happens here? Okay, a lot of floating things. Glycolysis into NADH, NADPH. You have, citrate going here, going back. Citrate. Wait, let me just type it here. Yes. Okay. Again, we have the citrate going back there, and then it's also remade into oxaloacetate, and then and then acetyl-CoA here, fatty acid. This is where see they're all connected, the fatty acid synthesis. So you now see glycolysis, TCA, ETC somewhere over there, and then fatty acid synthesis here. And at some point, squiggly line to malate, malic enzyme back to pyruvate. So they are all interconnected. All of them. Yes. Oh, by the way, if you don't like your hat, you can type exclamation hat like this. Oops, not the capital letters. Like that. And you can change your hat multiple times. Overall reaction, we talked about this, so that's fine. Let me just leave a mini, what's this called? Mini sticky note. So that we talked about condensation. Ooh, that's okay. Palmitic acid, you need a lot of cycles, but that's fine. Elongation and desaturation of palmitic acid. Well, cholesterol, not yet. So palmitic acid, you have the linoleic acid and linolenic acid. They're essential fatty acids because they cannot be further desaturated into humans. Yes, there's so many little dudes. It's so funny and bouncy. This is the first time this happened, so thank you very much for the mini chats, because it's like a mini class. Like, I was gonna say classroom of the elite or assassination classroom, but I kind of don't want to be Koro Sensei at this moment. Classroom of the elite is much better. Yes, the big brain topics. No. It could be the big brain topics or the simple topics, because this all started with we ate steak or a burger. What happens to it? You chew it, you break it down. Mechanical chewing goes to your stomach. Stomach will now have the lipase and will break it down even more. It's all about breaking down. After that one, it goes to your small intestine, that is where it gets absorbed, and now it goes to your bloodstream. Let's count how many little dudes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yay! Yeah. After it is in your bloodstream, it gets delivered to the different parts of the body. So yes. Now, let's move to cholesterol, a much more familiar word that we know. Cholesterol, we always hear it in ads, in our dreams, everywhere, in our books. So why not draw it? Let's draw it out. So cholesterol. It looks like this. One molecule of it looks like this. What is that? Wait, I want to draw it properly. A little bit properly. Just so it looks proper. Yup. That is a 6 ring. There you go. And we need another one here. Yup. And then we have 5 here, like a house. You have a methyl here. You have 1, 2, 3, 4, 5, 6, 7, 8. Another methyl here. Oops, nope, not there. Wrong side. Okay, here. Yep. Double bond. And an OH. But don't worry, I think it sounds, what is it? Sunday, 7 a.m. or 8 a.m. JST. But EST PST would be Saturday evening. I will do Power World. So that is where we will do Power World. Whoop whoop. I will release a schedule once I've thought of the thumbnail. I just have to wait until the thumbnail strikes me. It's like, what's a funny thumbnail today? So cholesterol comes from 2 main sources: dietary intake and de novo synthesis, with 50% from each. And you have the tissues like your liver and intestines and adrenal cortex and reproductive organs. They're capable of synthesizing cholesterol. Note, the liver and intestines are the most metabolically active in terms of cholesterol production. So, cholesterol synthesis, where does that start? It all starts in the cytosol, okay, and continues to the ER. But its carbon source, acetyl-CoA, is from the mitochondrial matrix. The thing is, acetyl-CoA cannot directly cross the inner mitochondrial membrane. And thus, to overcome this, first you convert it to citrate. Okay? Convert it to citrate, and then you export it to cytosol, and then back to acetyl-CoA. It's kind of the same thing from before. Convert, then reconvert. Yas. So, cholesterol. We talked about the synthesis of cholesterol. And now, how is it regulated? There is a thing called HMG-CoA reductase. And that is the primary step. It reduces HMG-CoA to mevalonate. It's inhibited by its product and also its end product, the cholesterol. And if you drink statins, like medicine such as Lipitor, that targets the HMG reductase. Okay? It is a target of statins. Some of the stuff that you drink, or your parents drink, or grandparents, for their heart. And the Lipitor would mimic the mevalonate, or act like it is, and competitively inhibit the enzyme, which would reduce cholesterol production. Okay? Because we don't want more cholesterol if you already have a lot. Cholesterol is not broken down for energy. Instead, it is removed from the body by excretion in bile, or through bile. Okay? And to summarize, just remember the mevalonate pathway and how you need to get rid of extra cholesterol. But it's not all that bad, because cholesterol also helps your membrane be like more fluid and not be too rigid. But of course, if you have too much of it, then that's it where it is bad. Note, cholesterol is also a precursor for vitamin D, because in the skin, the 7-dehydrocholesterol is converted. Let me write this. So here you go. Cholesterol is the precursor for vitamin D. There. You have this is the 7-dehydrocholesterol. It's converted by UVB light into vitamin D3. So that's what they mean by you need sunlight. Just think, you are also a plant. You are not a plant, but you get what I mean. You need water and plant and light. So go outside, maybe touch grass. I will try to touch grass. I don't want to, but okay, fine. I'm just looking at this now. It seems like I do need light to convert some of the 7-dehydrocholesterol into vitamin D3. Okay, and that's why you need the sun as well. We can't hide forever. Sag. Okay, so why does fatty acid synthesis occur in the cytosol while beta oxidation takes place in the mitochondria? Well, that is for independent regulation, and it prevents the random cycling of it. Because beta oxidation breaks down fatty acids to generate ATP, and it occurs in the mitochondrial matrix. Meanwhile, fatty acid synthesis, the making, is it takes place in the cytosol. Okay, so that's why, just so you don't mix them up. How do statin drugs lower cholesterol levels? They are competitive inhibitors of HMG-CoA reductase, which is the rate-limiting enzyme in cholesterol synthesis. And if you mimic the structure of mevalonate, they would bind to the active site and block the conversion of HMG-CoAs. Okay? So that's why you drink medicine. I mean, yeah, statins, if you need it. Now let's talk a little bit about the polar lipids. Yeah, we talked about a lot. Okay, you have the polar lipids, which would include glycerophospholipids, sphingolipids, and eicosanoids. And this is very different compared to like the TAGs, because TAGs are neutral lipids. Meanwhile, polar lipids have polar head groups and are particularly mostly involved in membrane structure or signaling. Okay? And due to their amphipathic nature, these molecules would be for the lipid bilayers that make up the cellular membranes. Okay, let's start with like glycerophospholipid. I'm gonna draw its structure. Glycerophospholipid. Glycerophospholipid. Okay, we have a structure. CH2O. Let's write it first. CH2O. CH2O. You have the C double bond R. C double bond R. And this is where the phospho term comes from, because it has phospho and an R there, or an X. This could be anything, okay? Well, anything that it needs. So that's a glycerophospholipid. CH2O, 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 COCR, OCR, OPOO. Okay. Hydrophilic heads and hydrophobic fatty acid tails. That would look like the one that we know. It's gonna look like this. It will look like this one. This one. This is a glycerophospholipid. Hydrophilic heads, hydrophobic tails. How do you know which one likes water and hates water? Okay. This is the ones inside. They're running away from water, so that means they hate water. That's the hydrophobic, of course. Phobia, scared of water. Meanwhile, the heads like water. There you go. So that's how it is organized. That is the glycerophospholipid. Phospholipids. Arrow. Yep. This provides the structural stability and promotes membrane protein functions. Whoop whoop. Okay, now we have groups 1 and 2. Oh no. Okay, let's write that out. Here. Lipids. There. Oh, not the purple one. So glycerophospholipids. Glycerophospholipids. Okay, now you have groups 1, G1, and group 2. Later. Group 1, you have ECS, ethanolamine, choline, and serine. And this would form phosphatidyl ethanolamine, phosphatidyl choline, phosphatidyl serine. This reminds me of what was the basketball team in Kuroko no Basket? Was it serine? Serine, one of those. I think it's a similar term. I don't know. I might just be imagining it. But yes, that is the common head groups. That's the part in the X that will replace the part in the glycerophospholipid. Meanwhile, we can jump to group 2. Group 2. Okay, group 2 is IG inositol-glycerol cardiolipid phosphatidylglycerol. Let's just write it like this: IgC phosphatidyl inositol phosphatidylglycerol, and then the last one is cardiolipid. So that's how it's just distributed. They have different ways to form themselves as well. And take note that you also have the Kennedy pathway, which refers to the de novo synthesis of phosphatidylcholine and phosphatidylethanolamine. A lot of things going on. And sphingolipids. Yes, sphingolipids, if we write it, it is also in like different groups. 1 second. Okay, if you have different groups of it, of course we have to write a map. There you go. So sphingolipids. You have sphingomyelin, sphingomyelins, and then you have sphingolipids, golipids, just different sugars and all that. Glycosphingolipids, that would have the cerebroside, sulfatides, globosides, gangliosides, just a lot more different things. Just remember sphingomyelins and glycosphingolipids, the sphingos. Oh, you have different storage diseases as well. Let's go with the sphingolipid storage disease, or sphingolipidosis. It is from inherited defects in the lysosomes, because if those are defective, sphingolipid would accumulate in the lysosome, particularly within the phagocytes that would normally engulf the dying or damaged cells, and the accumulation would cause neurological damage. So that's why it's much better to just have it checked, because it's like a genetic thing, and most are autosomal recessive and not curable. So handle that one with genetic counseling before pregnancy. Okay? There's also the clinical features of the different diseases. So foam cells, you have it. Those are lipid engorged phagocytes observed in the bone marrow or other tissues. You could also have something in your eye, like in your retina, caused by the accumulation of sphingolipids in the retinal neuron. And just remember the different groups. Today I learned genetic counseling is a thing. Yes, it is a thing. That you have to like have it checked up. How am I supposed to donate? Which one? For the genetic counseling, you mean? Or for the chat thingy? I think you might be able to see the mini square with a $sign inside. Yeah, the square with a $sign. I think that's how it works. But thank you. Yes. And then you have, thank you for asking. Yeah, the button is sometimes there. Eicosanoids, that's just another thing that happens. You have the prostaglandin H. Thromboxanes, I like thromboxanes. Those are produced by platelets. And those act, okay, remember, if you bleed and then it stops bleeding, that means your platelets acted, okay? They aggregated. And wait, anyone here watched Cells at Work? The little platelet thingies are so cute. I think those are the little ones, right? Yeah, they go, and then just block it. They would limit the bleeding. And then, what else? We have the leukotrienes, which is for allergic reactions and inflammation. Lipoxins, not as not those are not understood as much, but it's all right. Okay. So what's the Land's cycle and how does it contribute to membrane diversity? Well, that's just like the Land's cycle involves the acylation of phospholipids to form lysose phospholipids and other stuff. Different diseases, they differ in structure and function. And arachidonic acid, which sounds like a spider for some reason, it's considered the primary precursor for eicosanoids. Prostanoids, like the thromboxanes, which makes the why am I blanking on the word? The ones that block the platelet function. And leukotrienes, that's for leukocyte. That's the leukocyte recruitment. Okay, so now what did we learn so far? Fatty acids or triacylglycerols are energy sources. It's an energy source. Cholesterol, that's involved in membranes, bile salts, steroid hormones. Glycerophospholipids are for membrane components. Sphingolipids, membrane components. Eicosanoids, those are hormones. And then you have plasma proteins, which are for lipid transport in the blood. Okay? Lipid transport from intestine to tissues, you have chylomicrons. From adipose to tissues, fasting state. Liver to tissues, you have VLDL and LDL. Wait, this one's a good one, which I will bring up in a bit. Okay, so metabolic syndrome is a cluster of risk factors that we have to watch out, because that significantly increases the risk for cardiovascular disease and type 2 diabetes. So you gotta listen with this one. Oops, sorry. My alarm rang. Cardiovascular disease and type 2 diabetes. So, abdominal obesity, such as a waist circumference of greater than 102 centimeters for men or greater than 88 centimeters in women, and low HDL cholesterol, high triacylglyceride, elevated fasting blood glucose, and high blood pressure. Those are not a good combination to have, so maybe try to lessen them, because that will increase your risk of your heart disease, cockroach disease. Lipoproteins, you have the very low density lipoprotein, LDL, HDL, most dense, most protein rich. And then HDL is involved in reverse cholesterol transport, and they're exchangeable. I'm trying to find the page which says which one's the bad, which one's the good. Oh, wait, exogenous and endogenous pathway. Exogenous pathway is in the intestine that will produce the chylomicrons we talked about. Endogenous is inside the liver, producing VLDL. Let me just scribble that out. Endo and exo. Exo, EXO. Listen. Exo. Okay, exogenous pathway. Exogenous pathway, that is in the intestine, produce chylomicrons. And then you have endogenous, which is in the liver, endo. Yes. Later, it will produce VLDL, IDL, and LDL. What else? Liver, remember, liver is the only organ that can catabolize cholesterol and remove it from the body. So take care of your liver. Don't drink a lot. Okay? And yeah, you only have one liver. And I think it might be hard to get a liver transplant, because that would mean getting another liver from another human, which I'm not sure is feasible. I don't think they'll survive if they don't have their own liver. Okay, so just a mini good. Yeah, do not, just to make sure that your liver is all right. And make sure you don't drink a lot of painkillers, because that will also be rough on it. It's gonna make it work on hyperdrive. Intestine, we talked about intestine, the liver, and some other tissues. Okay, so from the liver, I'll just draw it. And this intestine is squigger. It doesn't exactly cause it, but if you drink, if you don't properly drink painkillers, like if you don't do the suggested doses, then that's a bit dangerous. If, for example, drinking painkillers more than 7 times or something, seven consecutive days or something like. Anyway, if you drink medicine that you don't exactly know how much you should be taking and how frequent you should be taking it, or if you just feel eternal pain, have yourself checked, just to make sure that you're not screwing up your own internal organs that might already be screwed up. Okay, anyway, just talk to your doctor to make sure that what you're doing is all right and you're good to go. So liver, bile acids, and cholesterols. Here you go. And then this is chylomicrons, and then you have the LPL and circulation, the chylo remnants over here, VLDL over here, circulation again, and LDL over there, and the extra tissues, LDL to the liver, and another arrow over here. See? All interconnected. Beautiful. Okay, LDL metabolism is critical because elevated LDL is strongly associated with what? Atherosclerosis. So that's why you need to metabolize LDL. But Type 1A milky white plasma, you see it in your blood. You have deficiencies, xanthomas. You also have inherited defects could cause hyperlipidemias, characterized by buildup of specific lipoproteins in the blood. You have type 3 hyperlipoproteinemia, which arises from mutations in ApoE. Okay. Plot twist, or what's it? No, pop quiz. Which one's the good one, HDL or LDL? HDL is much better. Okay, let's just HDL versus LDL. And let's explain why. The HDL is much better than LDL. There's a reason why I'm writing it this way. HDL is good because it's more compact and it can travel much better. Meanwhile, LDL is bad because this one's the one that could clog up your stuff. Okay. And what else? Oh, there's a thing called greetings, Sheriff Pumpkin. Thank you for coming to the stream. We were talking about the HDL and LDL and which one's the good one and which one's the bad one. The HDL is the good cholesterol because it will help remove excess cholesterol from the tissues and returning it to the liver. Meanwhile, LDL is the bad one. It'll cause the buildup that we're talking about. Transport. Lots of transport. Oh, what is the timeline of atherosclerosis? Let's talk about atherosclerosis. Here. How many of the floating things are here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I think 12 so far. That is nice. Okay. So you have atherosclerosis timeline. Atherosclerosis begins in early life with endothelial dysfunction. And then around the 4th decade, that's when you see the thrombosis, hepatoma, and all that jazz. And if you don't control it, they might lead to like rupture and some acute cardiovascular events. So please go have a check. Go talk to your doctor. Check your blood. In cholesterol transport, we talked about this. Oh, and the mini last part of the lesson is just the circadian clock and the lipid metabolism. And some people in 2017, Hal, Rosbach, and Young, they won the 2017 Nobel Prize in Physiology or Medicine for their work in the circadian clock, which also impacts the lipid metabolism. And they studied it in clock mutant mice. Those revealed hypertriglyceridemia, primarily due to the disrupted regulation of lipoprotein metabolism in the liver and intestine. And the circadian clock and lipid metabolism actually controls the genes in lipoprotein assembly and clearance. So that if you regulate your circadian rhythm well, that will help you with the feeding cycles and daily rhythm. So essentially, it'll just help you sleep better, apparently. Okay, so just a mini question. Why do lipids require specialized transport systems in the blood? Well, you have the short and medium chains and the long chains, so those just need specific stuff. And explain the concept of circadian rhythm and regulation of it in lipoprotein metabolism. They exhibit the genes involved in lipid metabolism exhibit diurnal expression. The SHP would repress the MTP during the day. Meanwhile, at night, it declines, which allows the lipid release, and any disruption on it would lead to hypertriglyceridemia, which shows the importance of time-dependent lipid regulation. Hooray! We finished lipid metabolism. Nice. So yeah, so far, maybe the next one might be nucleotide metabolism. But I think that is all for now so far. Thank you very much for coming by, and let me just drop the dab emote. Dab, dab, dab. Yes, thank you very much for coming by and learning so far. Just the mini lesson is take care of your liver, and maybe touch grass so that you can get some sunlight, and it will convert it into your some 7-something cholesterol into vitamin D3. So let's just count the head count right now. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Some of them are walking, so it's hard to count, but that is nice. So yes, thank you very much, and have a good, what is today? Wednesday or Thursday? Time zones, Earth. Okay? Thank you for watching. Bye bye.